And we're rolling. So I'm going to hit present, and there we go. Okay, plenty of stuff to cover. All right, I'll probably not get to this group called the Placozoa until Wednesday, but we'll finish up on the sponges, and then I'll start talking about cnidarians. This is purely a review. We showed you this on Friday. There's several different ways that you can, that sponges organize. And the simplest is Ascon grade, which is basically the sponge is a sac with a bunch of holes in it. And you have the panacoderm, the quote, outer skin, lining the outside, and a layer of choanocytes lining the inside. And water only passes through pores, through the walls of the sac, into the cavity inside, the spongiceal, and then ultimately out the osculum up at the top. I've actually got, and those of you that have kits, you can look at this yourself if you want to. But you've actually got slides of a sponge called leucoselenia. And what I've done, if everybody can see this, I've just put the leucoselenia slide on my own scope. And what you'll find is that leucoselenia looks like this. Can everybody see that adequately?
Yes.
Okay, great. Okay, Jamika, you got the right number. That's good. Okay, most of what you're seeing there is spicules. And in this particular species, leucoselenia, you're seeing lots of spicules there. And the spicules are all, the fancy word is triaxon triactine, which is another way of saying that they all look like the letter Y. That they're all made of 3 points radiating from the center. And I've got one kind of in the middle of the field of view that shows that pretty nicely. Right, they're kind of, jumbled together, and it may be a little bit tricky to see, but if I can find one that's a little bit better isolated, yeah, there's one in the center where you can see it. There's doodly doodly doo. Yeah, that's the one we just had. There, they're kind of sparse right there. And yeah, there's a couple that are kind of close to the center. As I focus up and down, you should be able to see that they're all made of 3 points radiating from a center. So they're all kind of Y-shaped. And again, that's typical for this particular species. Identifying sponges involves looking at the number and size and types of spicule. The other thing I wanted to show is that this is, in fact, a Leucon grade sponge, and it's not that easy. There, I've kind of focused on the middle of the body, and all of the green stuff, that's the living cells of the sponge. That's the actual sponge tissue, and all it is basically a tube. You've got the opening of the tube is kind of down at the lower left at about your 7 o'clock, 8 o'clock, that kind of blunt area with the spicule sticking out. That's just one of the openings of the tube. And as we try to follow it to another opening, it admittedly does get a little messy. I think this one's been torn. Here we go. That's a little bit denser area. All all you're seeing, all of that green stuff is just living sponge tissue, living cells, and they pretty much only make one, kind of sandwich-like layer. You have pinacocytes on the outside, choanocytes on the inside, maybe some mesohyl in between, but you don't really see anything branching. There's no passageways. There's no, there's no chambers or anything like that. What it just looks like is, just kind of a smooth green sheet with a whole bunch of spicules stuck in it. That's a cycon sponge. Leucoselenia is cycon grade. If you get the chance, you can check your own slides in your own time. There's a second one in here, and this one tends to be extremely annoying to view because they tend not to stain very well. This is a larger and more complicated sponge that's called Scypha or Grantia. I'm not quite sure why the thing has two alternate names. And this tends to be really annoying to look at because the staining is faint. It's hard to find. It's hard to see what you're supposed to be looking at. So I'm going to try to find this, even though I can't entirely see what I'm doing. And if I succeed, you'll get a nice view of a cycon grade sponge. If I said just a minute ago that Leucoselenia was a cycon grade sponge, I was clearly smoking toads. Leucoselenia is ascon grade. And this is a cycon, there we go. This is a cycon grade sponge. And I'll try to rack the contrast up as best I can. And what you're looking at is a sponge that's had its spicules dissolved because they have to do that before they make thin slices through the sponge, so you're not seeing spicules. And right up at the top here, right about in the center of the field of view as I've got it, is the actual osculum. That's the cavity into which everything dumps. And you can see as we go down from the osculum, you can see there's a central open space. That's the spongiceal. Surrounding it, you can see these kind of irregular circular areas. There's 3, maybe 4. Okay, now I can see 6 that are to the left of that open space. And those are chambers whose walls are lined with choanocytes. So the actual choanocytes are on the inside of those kind of irregular bubble-looking things. And water flows through those, through pores in the outside of those. It gets filtered by the choanocytes, and then it gets dumped into the spongiceal. And exactly, this is not easy to spot because it depends on exactly where the sponge has been sliced, but there are connections between those irregular, blobby things and the spongiceal. So if this is not perfectly clear, I'm sorry. I'm explaining it as best I can without any way to point. But dead center right now is the osculum. And then as we move the osculum up, you can see how it opens into this long cavity that I'm trying to keep in the center of the field of view. That's the spongiceal. And on either side of the spongiceal, you can see these open spaces that look like irregular bubbles. That's the choanocyte chambers. And those open right into the spongiceal itself. So what you've got is much more surface area for the choanocytes to work. They can filter more water and they can do it more efficiently. Let me see if I've got one more thing on this slide. That was a longitudinal section going. All right, this thing's going to drop out of focus for just a second. Bear with me. That was a section going along the axis of the spongiceal. What I'm looking for right now is a cross section cutting across the spongiceal, because this slide should have that as well. Okay, that's where we were. Right, let's okay, this is supposed to be a cross section. I think they put it at kind of a weird angle. But yeah, what you're looking at here is, okay, the big empty space in the middle. The way it's been cut, it looks kind of a big empty parabola. That's the spongiceal. You're cutting across the long axis of the cell. So that's the spongiceal. And to the side of it, you've got these choanocyte chambers. And right there, kind of starting down on about your 7 o'clock or 8 o'clock and extending up and to the right, that's one of the choanocyte chambers. And you can see, because it's been cut at the right angle, you can see directly where it's emptying into the spongiceal. And all of these chambers empty into the spongiceal. It's just not easy to see unless you happen to have cut it at exactly the right level. So I'm not sure how helpful this is or if I'm just making it worse, but let me cut back to the diagram that might make it a little bit slightly more clear. There. So, okay, so there's that Ascon grade. And then if we go up a notch to the Psychon grade, that's what that looks like. And you can see that instead of just having choanocytes lining the inside of the spongiceal, you have them grouped in these chambers, and water passes through incurrent pores, through incurrent canals to the choanocyte chambers. The water gets filtered there, and then the chambers dump directly into the spongiceal. So it's almost like the spongiceal has these kind of bubbles along all sides. And in a fumbling way, I was trying to show you that this is the way that Scypha/Grantia is put together. It's a Psychon sponge. And I'm going to try to do a lab tomorrow. It's probably going to involve having you look at pictures, at images that I have to try to take. But I'm going to try to take you through some of the anatomy of a Leucon grade sponge. And, well, we'll see how that works. So in a Leucon sponge, what you can see there is that the choanocytes in yellow make these chambers, and there's canals going in and there's canals going out. The chambers don't dump directly into the spongiceal. Water's carried in, filtered, and then goes out excurrent canals, and ultimately those dump into the spongiceal. The significance of all of this is that it allows the sponge to get a lot bigger. The largest sponge I know of is the Caribbean barrel sponge, which is big enough, it can get big enough for scuba divers to hang out in. And it's able to reach that size because it does that, because it's able to do this branching arrangement of canals with choanocyte chambers instead of the ascon arrangement, uh, which keeps the sponge from getting big. Okay, that was clear as mud. Let's keep going. Sponges are masterful at filter feeders, but there actually are a few that are carnivorous, and this is one of them. The carnivorous sponges are found in places like marine caves, which are dangerous habitats to try to explore. If you go scuba diving in a cave, the great problem is that if your air starts running out, you can't just rise to the surface. You have to find your way back. And given that you're not feeling gravity and light's not coming from any particular direction, it's extremely easy to get totally disoriented and unable to figure out which way is up or down. Yeah, cave scuba diving is very dangerous. And there's a whole bunch of critters that live in marine caves that have not been studied very much, but this is one of them, Asbestopluma hypogea. And it's actually holding on to the surface by some spicules. And then that's a bunch of spicules that are surrounding the body. The body is just this kind of glowing white lump. It's not really glowing, that's just the light of the camera. But surrounding the central body, you've got a bunch of very long extensions of the body. And those filaments are covered with little hooked spicules like Velcro. And little crustaceans get tangled in those long filaments. They get hooked. And as they're hooked, the sponge's amoebocyte cells, the amoeba-shaped cells that just crawl around anywhere they're needed, they migrate onto the prey and they digest it in a process that takes about 10 days. And there you're seeing a few of these carnivorous sponges with actual, little shrimp trapped on them. There's another one that's related, which I love. It's called the ping pong ball tree sponge. These actually get fairly large, and those extensions of the body aren't just extensions. They've got little spheres on the end, and those spheres are covered with little hook spicules that create this Velcro-like surface that traps any little crustacean that blunders into them. And then the crustaceans are very slowly and probably painfully engulfed and digested. And the amoebocytes, by the way, actually can carry nutrients to other parts of the sponge. So the entire sponge gets fed. So yeah, just in case you thought that this was make-believe, there actually are in fact sponges that do feed on Krabby Patties. They don't exactly look like this, and they don't eat quite as fast as SpongeBob does, but there are in fact carnivorous sponges that eat crustaceans. So yeah, I'm told the guy who created SpongeBob was actually a marine biologist, and occasionally you can find nods to realism in among the wackiness. Although, to be perfectly honest, I haven't watched enough of the show to point to any really good examples. My kid never did like it, I'm afraid. Right. Anyway. So reproduction, most can reproduce asexually. Any broken piece of a sponge can grow into a new sponge. Freshwater sponges can actually survive winters by producing these little buds internally, made up of a cluster of cells surrounded by modified spicules that make a shell. And these can sometimes be transported from one body of water to another. They can also survive freezing and then sprout a new sponge in the spring. And these bodies are called gemmules. And I'll show you one in a bit. Sponges can also reproduce sexually. Some sponges brood, meaning they retain eggs inside the body for a time and allow the eggs to develop until they hatch into larvae that can swim away. Other sponges are just broadcast spawners. which means they just fire off mass clouds of eggs and sperm into the water and trust to luck. So that's what the gemmules look like of freshwater sponges. That round spherical wall is made up of tightly packed spicules. And in the two pictures on the bottom, you can see this kind of slightly conical structure starting to grow out of the gemmule. The osculum is labeled. These gemmules were frozen and then they were thawed. And the whitish stuff that you can see coming out of the gemmule is cells of the new sponge beginning to organize into a recognizable sponge body. So this happens in freshwater sponges. If anybody gets the chance to, you don't have to make a special trip, but they're supposed to be found on the underside of rocks in some of the freshwater lakes around here. So if you get a chance to turn over some rocks, if you see a kind of jelly-like growth on the underside, there's a chance those are freshwater sponges. If you don't get a chance to go look, don't worry about it. And there you've got one of those Caribbean barrel sponges, genus Zestispongia. And if you look carefully, you can see it's got what looks like white smoke coming out of the osculum. That thing is just firing off mass clouds of probably sperm. Most sponges are hermaphrodites, but they'll only release one type of gamete at a time. And this is a great example of broadcast spawning, where sponges and other marine invertebrates do this too. They'll just fire off big clouds of gametes into the water and hope for the best. There's others where the sponges will retain their eggs in their tissues, pick up sperm by filter feeding, and if their eggs are fertilized, the eggs will hatch into larvae that have flagella on the outside and can swim away and then eventually settle down and turn kind of inside out, so the flagella are on the inside, and then you've got your larval sponge. So some sponges are brooders, but many are broadcast spawners, and you'll see both those terms again applied to other critters. Sponges, one more reason why all of this stuff might actually matter to anybody who's not a total geek, are that sponges are one of the current new frontiers in drug discovery, because I know I said this before, but it bears repeating. If you are an invertebrate and you can't move, and the fastest you can crawl is 4 millimeters in a day, you're going to get eaten if you don't have some kind of defense. And the spicules are a defense, but another type of defense is tasting bad, or better yet, being poisonous. So sponges, and very often it's the bacterial symbionts that actually do the chemistry, but they're a rich source of bioactive compounds, and many of them have antibiotic or antifungal or anti-cancer properties. And this is one places where people are looking for a source of new pharmaceuticals. There's a number that have been identified. Some of them are going through clinical trials. Some of them have been identified, but currently they're not practical because we don't have a cheap way to synthesize them, which is why you ought to pay attention in organic chemistry as well, because working out organic synthesis pathways, well, I hated it when I took OChem, but it's important. One that's already in use is a drug called Averol. That's a promising antimicrobial and anti-cancer drug. And because the sponge doesn't produce very much of it, until we come up with a way to synthesize Averol, you're going to have to grow a lot of sponges in order to get enough of a supply. Fortunately, Disidia avara, which you can see here kind of in the middle of the slide, you can see 3 Disidias with 3 osculums. They're all growing on a rope. The rope is held in the ocean horizontally. And this is one sponge that you can culture and then harvest Averol. So that's how they're getting enough to go through the clinical trials that last I looked, Averol was still going through. I'm going to pause for just a sec because I heard a ding dong. Okay, morning, Garrett. All right, I'll take it that he's here. So going back to the presentation, Garrett, we were just talking about sponges as a source of bioactive compounds. And this is one that's being cultured as a source of a promising antimicrobial and anti-cancer drug called Averol. I don't think it has met with full approval as yet. Last I looked, tests were still ongoing. But sponges in general are a promising place where people are starting to look for new antibiotics and new drugs in general. Right. Traditionally, we classified sponges into calcarea, which have calcium carbonate spicules that are usually all the same size. Hexactinellida, which have spicules of silica that are always hexactine, which means that they've got 6 points, right? 6, points coming out of the center, and the points are all at right angles. So you have 2 points facing up and down, 2 points pointing left and right, and 2 points pointing towards you and away from you. Hence, hexactinellids, 6 points or 6 rays. Hexactinellids have an organization called syncytial, and that's a word you're going to see again. And most hexactinellids live in the deep ocean. This is probably the group of sponges we know the least about. Far and away the most diverse group is the demosponges. They have spicules of silica, but spicules that are never hexactine. They've always got more or fewer points. The spicules are supplemented, and in the case of bathtub sponges, they're replaced by a network of fibers made out of a collagen variety called spongin. And this is 90% of all living species. And hexactinellids, you can tell right away because the spicules are being hexactine, they're all right angles, right? If you have 2 points pointing up and down, and 2 pointing left and right, and 2 pointing towards and away, all of the angles are right. And the hexactine spicules will often interlock to form these tight networks. And the networks turn out to have an awful lot of right angles in them. You can see how this is, some of the spicules aren't fitting the pattern, but most of them are interlocked into this right angle pattern. That's very typical of hexactinellids. They've got lots of right angles in their architecture. And hexactinellids are also syncytial. And again, that's a word you'll see again. What that means is that hexactinellids aren't made of separate cells. A syncytium is what you get when you either have one cell whose nucleus divides but whose cytoplasm doesn't. So it goes through mitosis but not cytokinesis. Or it's what you get when you have multiple separate cells that all end up fusing. Syncytium is basically a big blob of cytoplasm that's got multiple nuclei. There's one group of slime molds, the so-called acellular slime molds, that are syncytial. Veterans of parasitology, you might remember that the tegument, the outer skin of flukes and tapeworms, is a syncytium. We'll talk about everybody else. We'll talk about that later. That's coming up. And the entire body of a hexactinellid is a syncytium. So this is a word that you're going to see again and again. You might as well pick up on it now. And the cool thing is that there's a cat on the keyboard. Hello, cat. Please get off. Hi, cat. Hey. This allows the sponges to actually conduct electrical impulses quickly. Now, all sponges can actually do this. Every cell in a sponge can pass on an action potential to all of its surrounding cells, which means that if you poke certain sponges, you can actually track waves of contraction moving down the sponge because every cell can transmit an action potential. The problem is with most sponges, the rate of action potential transmission is measured in something on the order of inches per hour. They're not very fast at it. Hexactinelids can conduct electrical impulses much more quickly. I'm not entirely sure I know what the functional significance of that is, but we don't know that much about hexactinelids because most of them live in pretty deep water. They're not easy to study. That's the old way that we used to classify sponges. The latest seems to show that the porifera is actually paraphyletic. That demosponges and hexactinelids make up one clade that's shaded in slightly darker. Okay, never mind. The demospongia are actually down there at the bottom. The calcarea are actually more closely related to the rest of the animal kingdom. And there's one little group of sponges called the homoscleromorpha that actually show true tissues. In epithelial tissue, like your skin or the lining of your mouth, the cells are all budded together to make a layer, and they're all stuck to a kind of felt-like layer of extracellular matrix called the basal lamina. So not only are the cells lining the interior of your mouth stuck to each other, they're stuck to this kind of thin, felty backing layer of collagen and other fibers. That's the basal lamina. And the vast majority of sponges don't have that. The cells are stuck together, but you might remember that we can actually take the cells apart of most sponges. We can squeeze a sponge through a strainer and break it up into its individual cells. And most years I actually get to do this. And the cells will just kind of wander back together. The cells will make like the beetles and come together right now over me and build a new sponge. Sponges don't really have this kind of tissue level of cell integration. But homoscleromorph sponges, this is one of them, actually does have that basal lamina or basal membrane made of a particular type of collagen. It happens to be called collagen 4, underlying the panachoderm and the choanoderm. So these guys seem to be more closely related to the rest of the animal kingdom. They share a more recent common ancestor with the rest of the animals than they do with the other sponges, because they've got this unique feature that's present in all other animals but isn't present in most sponges. As we say, it looks like homoscleromorphs are what we call the outgroup to all other animals. So this is kind of cool. We've got some sponges that still preserve what looks like a very early stage in the evolution of more complex animals, the ability to form tissues instead of being a bunch of cells that are just hanging out. Okay, how are we doing? Have I completely confused everybody? Okay, it's 9:50. That's actually a pretty good place to take a break. I do not see anything coming in on the chat. Has confirmed this on the chat if you don't mind. Is oh yeah, Can everybody see the screen adequately? Am I right that my problems with my monitor, they're not affecting you, are they? Everybody can see clearly that I've got this very annoying black and white cat right in front of me because, of course, I always get photobombed by cats. Yes, you are such a nuisance. Okay, Autumn says yes. Okay, Jamaica says yes. All righty. I'm going to go ahead and I've got 9:51. Come back at 10. Go ahead and get coffee or do whatever you need to do. We'll take a break. We'll come back at 10 and I can move on to jellyfish. So off the camera and turning off the mic for now. See you guys in about 10. We're back. Um, what do they used to say? We're back. It's Click and Clack the Tappet Brothers, and you're listening to Car Talk. Or I guess this would be Invertebrate Talk. No, Invertebrate sounds like New York. This is Boston. Invertebrate Talk. Sorry, can't do accents this early in the morning on one cup of coffee and really shouldn't try. Okay, right. We are commencing. I just realized